All right, back to the immune system. So in this video, what I'm planning to cover is the rest of the innate immunity. So just to look at the outline, last time we covered the second line of defense. These are non-specific internal defenses. If some kind of pathogen gets through the first line of defense, skin and mucosal membrane, the cells and the chemicals that we talked about last time come into play. And in this video, I want to talk about the other two things that are also considered. These are two processes that are considered part of your innate immunity, non-specific immunity to pathogens. So the main process is number 3 here, the inflammatory response. And that's what we'll spend most of our time talking about. But fever is also considered to be an innate immune response. There are beneficial effects of a fever, especially a moderate fever. All right, so let's get into the material. So for the inflammatory response, also known as inflammation or tissue inflammation, a general description of what goes on. So it is a local response to a number of things. It can be infection or overuse of the body. For example, a pitcher pitching a long game will have inflammation of shoulder or something in the pitching arm. Tennis player, same thing. Overuse, infection, or to some kind of agent such as a bee sting or something like that. Okay? Inflammation is non-specific, local, and it's a very rapid occurring immune response. So all of those are features of innate immunity. And it does occur in vascularized tissue. So the blood vessels feeding a tissue are a big part of what happens in an inflammatory response. So you sometimes hear about inflammation as being problematic. There are all kinds of drugs to treat inflammation, but that's only in cases where it's a chronic type of inflammation or an inappropriate type of inflammation, which can interfere with normal function. The inflammatory response is, in fact, a helpful response of the immune system to control or slow down an infection and eventually to help get rid of it from the body. What are the steps in an inflammatory response? So there are 3 of them. The first 2 are shown on this slide, the third one on the next slide, and then the following slide will have an illustration of these 3 steps. So the first thing that happens is either there's tissue damage due to overuse or an injury, or there's an infection, or both. These things cause a release of a storm, a huge amount of what are called pro-inflammatory chemicals. These can be released from the tissue cells themselves, if injured, or from the basophils and mast cells that we talked about in the last video. Finally, inflammation can be triggered by an infectious agent, like a bacterium or something secreted from a bacterium. The chemicals that are released, the pro-inflammatory chemicals, include a long list of them, some of which are listed here: histamine, leukotrienes, prostaglandins, chemotactic factors. So some of these are contained within these cells, and if the cell gets injured, ruptured, they get spilled. Some of these are in the membranes of these cells, and they get released as a result of injury or infection. Okay, so step 1 is a release of a huge mixture of previously contained chemicals. These pro-inflammatory chemicals are released locally at the site of injury or infection. Second step, what do these pro-inflammatory chemicals do? So these have an effect mostly on the vessels in the local tissue. Okay? If you have damage in a tissue or infection in a tissue, the chemicals get released, pro-inflammatory chemicals. They're going to affect local nearby arterioles, and they're going to cause vasodilation, right? And they're going to affect local capillaries in capillary beds. And what they do in the capillaries is two things. One is the capillaries become much more permeable. So if they were tightly closed before, now there's lots of space between endothelial cells and the walls of the capillaries for stuff to exit. But this is only at the site where there's tissue injury or infection. It's a local response, okay? The other thing that's going to happen in capillaries is the endothelial cells making up the wall of the capillaries are stimulated to produce a molecule from the inside of the vessel wall that's called a CAM, cell adhesion molecule. So this molecule sticks out into the bloodstream like a tether or a piece of Velcro. And if a leukocyte, a white blood cell, bumps into it, it's going to latch on to that white blood cell. Okay? So those are the two main things that are happening to blood vessels: vasodilation of arterioles and increased permeability of capillaries, and also endothelial cells making cell adhesion molecules called CAMs. All right? Vasodilation is going to deliver more blood to the site. That's a good thing. It's going to deliver stuff that's needed to fight an infection or to repair tissue damage. And not only is it going to be delivered, it's going to be able to get out into the tissues because the capillaries are more permeable. The third step is what's called leukocyte recruitment. What this means is leukocytes, white blood cells, are recruited to the local site of tissue damage or inflammation. And we want that because white blood cells are going to battle infection, right? We talked about some of the white blood cells last time. The phagocytes, basophils, eosinophils, neutrophils. They're all going to be attracted to the site. How does leukocyte recruitment occur? Well, these three steps. Margination. This is where leukocytes are going to stick to the cell adhesion molecules on the inside of the capillaries, the CAMs that I just talked about. And margination is going to pull leukocytes from the blood in the circulation to the margin or the wall of the vessel. And then they're going to be tethered there, okay? Otherwise, if these cell adhesion molecules weren't there, the leukocytes would just flow right through, okay? But now they're stuck to the margin of the vessel. Second step, diapedesis. What this means is a white blood cell that's stuck to the wall now is able to squeeze between neighboring endothelial cells and wiggle out of the bloodstream and into the interstitial space. Diapedesis is exiting from the blood to the tissue. Finally, step 3, chemotaxis. When a white blood cell exits out of the blood to the tissue, it's going to follow a chemical trail, the trail of pro-inflammatory chemicals. From area of low concentration, let's say near the blood vessel, to an area of high concentration. This is the site of tissue damage or infection, right? So it's going to move up this concentration gradient of these pro-inflammatory chemicals, and that's going to bring it to the site of tissue damage or infection. So this is called chemotaxis. Chemo means chemical, taxis means movement. White blood cells are going to move up this chemical trail. Okay? So, those are the events in inflammation. Let's look at what it looks like on this figure. And they're numbered here, 1, 2, 3, just like in the previous 2 slides. Here are some leukocytes flowing through a bloodstream. Here there's no tissue damage. Here there's a cut. The guy skinned his knee. And the first line of defense, the skin, has been breached. And so bacteria can get into this, cause an infection. So this is causing both tissue damage and infection. Cells here are going to release pro-inflammatory chemicals. So all of these dots here are pro-inflammatory chemicals. Damaged cells are going to do this. Mast cells and basophils are going to do this. And even some of these infecting bacteria are going to release chemicals that are pro-inflammatory. Pro-inflammatory chemicals are going to cause local arterial vasodilation. They're not really shown on this slide, but that's going to increase blood delivery to the site. They also increase capillary permeability. So here, the spaces between endothelial cells are more wide open than they were over here. Okay, that's going to allow more stuff to exit. Fluid and also cells. Step 3 is the cells exiting. Leukocyte recruitment. We want to recruit leukocytes from the bloodstream into the tissue so they can do battle with infecting agents and help with the cleanup. All right, so step 1 is margination. Here is an endothelial cell producing a CAM, protruding into the bloodstream. You don't see that on endothelial cells up here, only in the area of tissue damage or infection. These are going to latch onto circulating white blood cells, and it's going to pull them to the margin. So this is margination. Diapedesis is when the white blood cell squeezes out of the bloodstream into the tissue. Step 2 is diapedesis. And then the next thing that's going to happen is this white blood cell is going to follow the trail of pro-inflammatory chemicals to the site of infection. Okay, so those 3 processes are going to increase blood delivery to a site, and it's going to allow for a lot of stuff to exit to the site out of the bloodstream. That's what we want. We want a lot of stuff exiting. Cells and also fluid and also proteins are going to exit. All right? So those are the 3 main steps in the inflammatory response. Okay, so not only are the leukocytes going to exit, but other things too. So one of those are a set of plasma proteins. They're present in the bloodstream. If you make the walls of the capillaries more porous, plasma proteins are going to exit too. What's the role of plasma proteins? So included in the plasma are a number of proteins that are going to help battle an infection. And those include immunoglobulins, which are another name for antibodies. Those can bind to bacteria and other things that shouldn't be there and help immobilize them. Complement proteins, like we talked about in the last video, are extremely helpful in battling bacterial infection. So they're going to opsonize bacteria and other things that we talked about last time. Also, clotting proteins are going to exit. You might not think of clotting proteins as being helpful, but what they do is they can wall off the infected area. Just going back here, clotting proteins can form this kind of protective wall around this infected area. And that's going to limit the spread of any infecting bacteria, right? Just because it's difficult to get through this sort of clot wall, okay? Also, you have excess fluid exiting from the bloodstream. So that fluid is called exudate. Exudate is the fluid that was in the blood vessel that's now exited into the tissue. You have a lot of fluid exiting because the capillaries are so porous now. So what is the role of an increased exudate during the inflammatory response? Well, one thing is it carries stuff that are needed for tissue repair and defense. One thing is the proteins that I just mentioned. but it's got immune system cells and also sources of, I guess, energy, so glucose and things like that to power the process, ATP as well. Okay, so that's important to help to battle an infection. You have to not only supply the materials, but the energy for them to do their thing. Another thing that happens is more fluid exiting from the bloodstream into the interstitial space is going to increase the swelling of the tissue. In an inflammatory response, there's swelling. So the swelling is actually a good thing. This raises the fluid pressure in the interstitial space. What does that do? Increased fluid pressure is going to pull open the flaps in the walls of the lymphatic capillaries. And you're going to have a lot of fluid being pushed into the lymphatic capillaries. It's going to flow away through the lymphatic system. And where is it going to go? Well, the lymphatic capillaries are going to lead to collecting vessels with lymph nodes along their pathway. So lymph nodes are sites of immune surveillance and immune defense. So the increased hydrostatic pressure in the interstitial space is going to be sucking in any of these infectious agents like bacteria and viruses and send them to a lymph node where they can be dealt with. If they're not dealt with at the site of infection, then they will be dealt with in the lymph node. Okay, so that's beneficial. Right, and that's shown here on the same slide. Plasma proteins being delivered, that's part of the exudate. And the fluid is mostly going to be doing this, exiting from the bloodstream. It's going to be being sucked into the lymphatic capillaries, and it's going to drag along anything that's in the interstitial space. Remember, what enters lymphatic capillaries is anything in the interstitial space. So in this case, it's a lot of infectious agents, bacteria, and maybe some damaged cell particles, right, sucked into the lymphatic vessel, capillary, and being sent to the lymph node. All right, how does inflammation end? What happens when the process begins to peter out? So in an inflammatory response, typically this gets resolved within 3 days or so, and the signs of the inflammation are starting to disappear. So what happens as a result is two things. The tissue will be cleaned up, and repair of the tissue is going to start or take place, I guess. Cleanup involves attracting a lot of monocytes from the bloodstream into the tissue. Monocytes, when they exit the bloodstream, become macrophages. Macrophages are the main workhorses, phagocytic cells, to clean up a tissue. So any kind of infectious agent still hanging around is going to be phagocytized. Any kind of cell damage also being taken up by macrophages. It's going to be patrolling. You're going to have a lot of macrophages patrolling this tissue and, cleaning up the any kind of debris or any remnant pathogens. Okay? Also, repair is going to happen. So there are cells from neighboring undamaged tissues that are going to migrate in and divide. These are the fibroblasts, and they're going to start repairing the tissue. And so they're going to lay down collagen fibers and kind of stitch up the tissue. If the epithelium is damaged, that can also be repaired simply by cell division of epithelial cells. And the end result is the debris is cleaned up and the tissue is repaired. Now, there are 5 typical, or what are called cardinal signs of inflammation. So I want to go through those just to mention what you should see or what you would see in an inflammatory response. Remember, this is a local response. So if you get a cut on your arm and maybe it gets slightly infected, you would see these 5 signs. All right, redness is the result. Why do you see redness? Well, because of the changes to the capillaries and the arterioles. More blood is going to be delivered to the site, and it's going to exit into the tissues. More blood means more red blood cells. More red blood cells means more hemoglobin. Hemoglobin is pigmented, so you're going to have more pigment at the site. It's going to be redder for that reason. It's going to feel hot. There's going to be heat at the site. This is partly due to the increased blood flow. Carrying blood to the surface of the body is going to be, bringing warm blood from the core to the surface, more heat. But also because there's a lot of enzymatic activity going on in battling an infection and in cleaning up and repairing any kind of tissue damage. Okay? There's got to be swelling. That makes sense because of the increased exudate being present at the site. So there's swelling in the interstitial fluid because of the leakiness of the capillaries. There might be pain, and the pain would be probably the result of a few things. One is maybe if the swelling is excessive, there is pressure on the site, and so compression of the tissue can produce pain. But also pain can result from chemical irritation due to pro-inflammatory chemicals, or also chemicals released by bacteria, such as toxins and things like that. Fifth cardinal sign is not occurring in all cases. So in some cases of inflammation, there can be loss of function. For example, if you have inflammation of the joints, then you can lose, at least temporarily, function of that joint. All right, and so these cardinal signs are illustrated on this flow chart of inflammation, showing what starts it all, some kind of tissue injury or infection, release of chemicals, histamine and kinins and things like that. And these are going to cause vasodilation of local arterioles, increased permeability of capillaries, CAMs on endothelial cells of capillaries attracting leukocytes. Capillaries are going to leak fluid, which is the exudate, into the space. Vasodilation produces hyperemia. I didn't define that term before, but you do need to know that. Hyperemia is an increased blood flow to an area, in this case as a result of vasodilation of arterioles. Hyperemia is going to cause heat, increased blood flow, and redness. Increased exudate is going to cause swelling and pain. Swelling might cause temporary loss of function in some tissues anyway. You can kind of go through the rest of this flow chart. It's related to processes that we talked about in earlier slides. But it's a good summary of what's going on in general. All right, the other process involved in the innate immune response is fever. You might not think of fever as an immune response, but it actually does have beneficial effects. First of all, what is fever? So fever is defined as an abnormal elevation in body temperature, and technically above 1 degree, at 1 degree over normal body temperature. So 38 degrees centigrade or above. Inflammatory responses can trigger fever, so that might go hand in hand. Now, somebody who has a fever, and this is more so for a higher fever, the higher temperature creates a higher fluid loss, just because more water at a higher temperature is going to evaporate from wet surfaces through breathing and things like that. So somebody with a fever needs to take in lots of fluids. That's the advice for a fever. Drink lots of fluids in order to replace lost fluids and prevent dehydration. What causes fever? So there are chemicals that can be released in an inflammatory response or from an infectious agent that are called pyrogens. Pyrogens are chemicals that cause a fever. So these chemicals, what they do is, depending, it doesn't matter really how or what their origin is, but when they're released, they will enter into the circulation and eventually they will circulate to the hypothalamus. The hypothalamus is the body temperature control center of the body. So normally it sets body temperature at 37 degrees centigrade, 98.6 degrees Fahrenheit. A pyrogen will cause the hypothalamus to reset body temperature at a higher temperature. Now, if you have a relatively low fever, so this will be in the range of 37 to 38 degrees, okay, or 98.6 to 100 degrees. So this is an elevation of temperature, maybe not technically called a fever, but a slight elevation in temperature. This is considered to be beneficial for all of these reasons. A slight fever inhibits reproduction of pathogens. It promotes the activity of certain innate immune proteins like interferons. And so that will help with adaptive immunity in general. It also promotes tissue repair. It increases the expression of cell adhesion molecules on the capillary endothelium in an area of tissue damage or infection. And so for these reasons, a low-grade fever is considered beneficial, and the recommendation is to not treat a low-grade fever. On the other hand, a high fever causes certain problems and is risky. And such problems include, first of all, definition of a high fever. So 100 to above 100 degrees Fahrenheit or 38 centigrade. And these can be dangerous above 130 in children or even lower in adults. The danger is that high temperature starts to affect protein structure in the body, and it can denature proteins. A denatured protein loses its usual shape, and it becomes at least less functional and perhaps even non-functional. So this affects metabolism. If enzymes are not functional, metabolic pathways are disrupted. And higher temperatures, higher fevers are going to produce even more severe effects. So the recommendation for a high fever is to treat it. And so Tylenol, ibuprofen, something like that, which will bring the temperature down. At extremely high temperatures, above 106 degrees, for at least a somewhat prolonged period of time, there can be the cause of brain damage. Above 109 degrees, likely death. Okay, I'm going to end this video here. So in the next video, we'll start talking about adaptive immunity. See ya.